are finally starting with Kingdom Animalia. We get to talk about animals.
6 kingdoms, right?
5 or 6, yeah, whatever. Yeah, right. We're talking about animals. Okay, let's just go through a little bit of review. This is not in your book. I just want to review it real quickly. When we're looking at comparison of the kingdoms of domain eukarya. All right, so we're not talking about bacteria at all, right? Because they're in domain bacteria or domain archaebacteria, right? So we're looking at domain eukarya. These are all eukaryotic cells. So in all of them, in the kingdom Protista, fungi, plantae, and animalia, they all have eukaryotic cells. What about cell walls? Cells lack cell walls. Some Protista, few fungi lack cell walls. None plantae. In other words, all plants do have cell walls. And animals all lack cell walls. Okay, no cell walls. That's one of the main differences between animal cell and plant cell. Multicellular, some, most, all, and all. So all of the animals we're looking at all are in Kingdom Animalia. They're all multicellular organisms. And then heterotrophic, we have some Protista. We have all fungi. In other words, some of the Protista can be autotrophic, can make their own food through the power of the sun or chemosynthesis. All fungi need to get their food source elsewhere. Remember, they're saprophytes. And then plantae, of course, none, because all of them, except for that weird Indian pipe, right, that doesn't go through photosynthesis. The rest of them go through photosynthesis. Animals, all are heterotrophic. We have to get our energy from somewhere. Okay, here comes notes. 2 divisions of Kingdom Animalia. Here we go. We have vertebrates, and we have invertebrates. You've all heard these words before. I wish there was some way that we could make them different enough to help us remember a little better. But what you just need to know, if it's a vertebrate, it does have a backbone. It has a backbone.
I like they changed it to like vertebrate.
For invertebrate?
No, the vertebrate and like I think
I'd like them to come up with a different name, but it's invertebrate, and you just have to remember. Just remember a vertebrate has a backbone, and then you can remember the invertebrate doesn't have a backbone. Yes.
What about non-animal and animals?
Well, we're just talking about Kingdom Animalia now.
Well, they're all animals, but some of them don't quite look like animals.
Well, maybe so. Yeah. But we're not talking about protists now. We're strictly talking about multicellular animals. But today and in the beginning here, this first couple of modules, yeah, we're going to be talking about invertebrates. In other words, we're going to start with the ones that do not have a backbone. All right? Because they do not have a backbone, they don't get very big. All right? Because their skeleton is on the outside.
Jellyfish?
That's true. They can get big. We're going to talk about those guys today. We're going to talk about that big one today. That's true. All right, so the invertebrates include creatures like this. Because they don't have a backbone, they typically have something else to give them some sort of protection, like a shrimp has that, like shell protection on the outside. So does a scallop, an actual shell that the creature makes.
Are these examples or no?
A sponge is an animal.
No, we're looking for symmetry types examples. That's not here yet. We're not here yet, okay? A sponge is an animal, yes. A jellyfish. A snail, remember it's got its shell on the back. Spiders. Starfish, these are all examples of animals that do not have a backbone. And the next couple weeks, next week we will be dissecting an earthworm. Classic, right? Good stuff, yes.
do we write this down?
No. And then the week after that we'll be dissecting a crawdad, I think, and then the week after that a grasshopper.
Called diddly.
Think so, yeah. All right?
No frogs.
All right, symmetry, this is what you're going to be writing down, the types of symmetry and an example of that type of symmetry. The first one is called spherical symmetry. Spherical. Okay, in something that exhibits spherical symmetry, you are going to see that if you cut through the center, both sides will look exactly the same. It doesn't quite go for top and bottom, but definitely through sides. So if I cut this open this way, it's going to look the same way if I cut it this way or this way. So an example of that would be a sea urchin. All right, I think eventually we're going to have a couple of sea urchins to look at too, just not today, okay?
Still working on that alligator.
Okay, so we're working on alligator. Cool, cool. All right, so what you can see right here, it definitely looks like a spiny ball right there, but it turns out that that spiny stuff is part of, let's say, the skin of the animal. Underneath it, it has some something called a test, or you call it a shell. It looks like that. All right, and so
cute.
you may have seen they actually where the spikes went in. This is where the spikes were coming out of it, and the tube feet were right along these edges right here, meaning they have holes in the shell. So sometimes when people find really, really big ones, they'll put a light bulb in there and make a lamp out of it, and they are kind of cool looking.
I didn't
Yes.
know they
What does
got
it feel
big
like
enough.
when you put your finger in the hole?
Well, that's just empty and say this is a dead one. Okay, but when we dissect one, you'll see. Yeah, the underside of it is one of the creepiest things you'll ever see. It's got
So I
this
can put my
massive
finger
mouth in
in the
there.
hole.
Yeah, well, we're gonna cut through it. There's body parts there. Yeah,
That's next week?
it's like putting your finger through an
No,
eye.
next week's the next one.
All right, another example, we've already looked at Volvox under the microscope, right? Remember they're living in a colony, but inside the colony are these little spherical things right there. That's another example of spherical symmetry that we see. The next one's called radial symmetry. Radial, it can be cut into identical halves on any longitudinal cut through the center, and it looks the same. Did you have a question, Stephen?
Yeah, you didn't give us an example for the Volvox.
It was spherical.
Part of the first one.
Oh, okay.
Yeah, part of the first one.
Come on.
The second type of symmetry is radial, and our example is going to be a sea anemone. And you can see anemone right here. You can see how it's cut through any plane right here, and they look the same. Once again, top and bottom, not the same. Okay.
I see it.
So they're similar to spherical, except they're not as round.
Like a weird octopus.
Yeah.
Spell it for me.
Bilateral symmetry,
and-
the vast majority of animals we look at will probably exhibit bilateral symmetry. That means you can cut them in half, like humans, for instance. If you cut me in half this way, I have an eye, ear, half a nose, half a mouth over here. Eye, ear, half a nose, half a mouth over here. Arm, leg, leg.
But
So
my
humans
heart is
exhibit
here.
bilateral symmetry.
Butterfly.
It's very general. So they have sort of a identical halves on a mirror, kind of a mirror image kind of thing going there. A butterfly is an example. You can name pretty much any other animal: dog, cat,
Alligator.
snake. As
I'm
long as you
assuming
come
these
along
are
with-
gonna be something that'll probably end up on a quiz.
Probably.
Okay.
Yeah, I suspect.
So we should tattoo it on our arm now,
Hmm.
and then tell you during quiz time, I'm so sorry, it just stayed there.
Tattoo, permanent.
Can't get it
yeah.
off.
All right, here's a blank you're filling in right here. The area of an organism that has concentrated sensory organs is called cephalization. Cephalization, meaning the brain and head are together, and that's where the majority of senses are processed. If they're not found there, like for instance, the sense of touch we have in our skin, but our brain interprets that. Yeah, what's happening?
Those are
Oh, the lid is stuck? Oh, wow, okay. All right, we use the terms anterior and posterior. In my biology 2 class, when we're talking about A&P, which is anatomy and physiology of the human body, they have to remember this all year. Anterior and posterior. Anterior is the front, posterior is the back. We also call it dorsal and ventral. Dorsal would be the back. Think of dorsal fin on a shark. Ventral would be belly. So sometimes I'll tell you that we're going to make a ventral approach on one of our dissections, and that means you're going to cut through the belly. Okay, so that's typically we cut through the belly on a animal dissection. We're going to dissect a grasshopper.
Can I
Can I cut the head?
They're dead already.
Can I cut over the head?
Absolutely.
So we're like great.
Grasshoppers are about this big. They're pretty big for a grasshopper, but they're not that big in general, right?
There's
So.
There was some at the tree farm. The biggest one we measured was 6.5 inches long.
Wow. Bring me some of those.
It's huge.
Yes.
I mean, I
We
could try.
dissected an ant.
An ant.
Yeah.
They're really hard to dissect. They're too small. So the answer's going to be no. Maybe you could smash it and then look at it. Yes.
We dissect my ant.
I'm you anti? That would be murder. So no, we're not going to do
What
that.
if they're already dead?
She said.
That would be great,
Also,
Robbie.
not going to do that.
No, it wouldn't.
All right,
Not-
Kingdom
not bad.
Animalia is very diverse. That means we have lots of different animals that fit into this group. We have all these different guys here, right? And so these would include invertebrates and vertebrates right here. We can see that line, I mean a tiger and a shark and a monkey. Those are our vertebrates, our invertebrates. Right along here. Yeah.
The monkey's invertebrate.
All right, we're going to talk about sponges a little bit. I like sponges. They are animals. All right, so it's called phylum Porifera. That's the word, Porifera. You need to remember that. I want you to remember because it says pore in the front of it. Pore as in P-O-R-E. Okay, so in other words, they have pores in them. They have holes in them. Doesn't a sponge have holes in it?
Yes.
Yeah, so this is a natural sponge right here. You see a bunch of holes in it. Okay,
Can
those
I
are the
eat it?
pores. No, they're not edible.
You're the
like
kid
cotton
at the
candy?
pool that bent
No.
the pool noodles, weren't you?
They are a natural pump. Okay, they're not plants. They're animals. They have no symmetry. So if it asks you for type of symmetry, write none. For symmetry of a
Wait.
sponge. You supposed to write that? No.
They're not plants, they're animals.
They have shapes that are flat, tube-like, branched, cup-like, vase-like. They're very, very diverse in their shape. Yeah, phylum Porifera. The sponges. Did you write that in that blank? A natural filter, right? Did we say the word? Was it filter?
It wasn't supposed to go back. Go back.
A pump, natural pump. Okay, and now you're going to say the anatomy, feeding strategies, special structures, and reproduction. So under anatomy, say no symmetry. They're flat, they have tubes, they have cup-like, vase-like parts. That's their anatomy. This is talking about the anatomy.
Flat.
Yeah, I'll show you a couple of examples of them. They're pretty cool. I like sponges.
Can we eat those?
Do you say vase or vase?
Who says
I say vase.
stupid?
It's vase.
It's VAs. I know.
All right. They have two layers of cells. The epidermis is the outer. Now I have those underlined, but you don't have that, right?
No, it's just.
Okay.
We have feeding strategies.
Okay, the epidermis, the endodermis that are separated by a jelly-like substance called mesenchyme. We're going to get that at the end. We're going to label that, so don't worry about it right now. So you see what happens here, those pores on the side. When we're talking about the side of an animal, that's called the lateral part, right? So so they bring the water in laterally, and then they pull it through those pores, and they take it out of the top. So they're pumping water this way and out of the top, and they actually end up cleaning the water. All right, so it pulls it through there, and all the junk that's in there is usually stuff they can eat, they can digest. So they'll pull that in through these specialized cells right
What do
there.
you mean by junk?
What we might call it, like it looks murky, the water looks murky, but really it's like a bunch of little zooplankton or phytoplankton.
So that's
They're
what
eating
makes it like the water clear and stuff.
Yeah, because they eat it.
So then, in like Matagorda, what's the place called? The place in Texas where the water's like crystal clear.
Paris?
Is it Matagorda Bay?
Yeah,
That's,
Matagorda, yeah, Matagorda. So
Yeah.
do they have a ton of these in the water?
I don't think we have much sponge action going on in the Gulf
So then, why is the water so clear in Matagorda?
because there's mangrove swamps there. Mangroves definitely clean the water as well.
I have a question.
Yeah.
Hypothetically, if someone had a pool and they put this in, would it?
The chlorine would kill it. The chlorine.
Let's-
The chlorine would kill us.
let's
But there are
talk
saltwater
about-
No
pools.
chlorine in
How
it.
about a
Our
saltwater
saltwater
tank?
pools.
A saltwater tank, people have sponges in there, and it
Okay.
does help clean the water. Absolutely.
Could you put it in a saltwater pool, do you think?
Probably. I don't
If you don't
know.
put chemicals, it's just salt and water.
Yeah.
It just has a filter.
They would
that
have to
runs
have
through.
some sort of source of food. In a tank, they would have the other fish who are going through their metabolic processes,
You could have
and
a
they could eat that.
giant
You'd have to have fish in the
at-home
pool.
ocean
Yes.
pool.
If you were to keep sponges like how you keep a fish, and like fed it, could you just keep it as like a sponge pet?
I suspect you'd have to get the salinity just right, the salt in the
If you
water.
did-
It's
like
pretty
say you
boring
did,
pet.
could you
That's
keep
the
it
hard
as-
part about a saltwater tank, keeping the level of salt. At
If
the
you
right
did,
level.
you could just keep
So
it as
So
a
like I
sponge.
have right here in this little jar, and y'all can take a look at these in a minute. I have some critters in here called sea monkeys.
See?
Oh, yeah.
Yeah, and those would probably feed a sponge.
That's you.
Yeah.
But do you magnifying glass so you can look at them?
Yeah, you can see them swimming around there.
sea
You, I mean, you don't even need a
I am
magnifying
not a
glass.
sea monkey.
Oh, how big? They got big.
They're getting bigger and bigger, yeah.
Nothing right
Okay, so we're gonna look a little more closely here. So think of this as like a sandwich, right? We have the inner part and the outer part, right? And in this between part is this really cool stuff. All right, so first of all,
Ridiculous.
many sponges have a skeleton-like substance in there, giving them, that's how they're able to stand
Oh,
up
you're
really
right.
tall like those tube ones. I don't think so.
No.
This might be you have interesting special structures. Under special structures, go ahead and write down this word. It is pronounced spicules. Some people do pronounce it spicules, and if you want to do that to help you spell it, that's fine, but it is pronounced spicules. We'll be looking at sponge spicules under the microscope today,
Can it be pronounced spicules?
if you would like to. That spicules is the way it's pronounced. Some of them are needle hard structures made of calcium carbonate, which is the same thing that shellfish make their shells out of. Okay, it's sort of chalky. They're not soft. They're kind of spiny if you touch them. I have several samples over here. These are what I actually used for my marine biology class. Y'all are going to get to look at some examples of other sponges from there. For my marine biology class, I actually take cuttings off of those and find the spicules, and we look at them. I did not do that for y'all. We just don't have time. Some spicules are made of silica, so they're glass-like.
That's cool.
Okay, and it's very cool, and they will poke you. It's almost like a sticker burr. You know, you pick up a sticker, and that sticker burr gets you. That's a lot of times what a sponge will do. Spicules can have really interesting shapes. The ones we're going to look at, they're okay. But here are a bunch of spicules all together. Here you can
Looks like
see different types of spicules. These are really close up. This is using
Those
electron
are
scanning
cucumber.
micrographs. But look at all the interesting stuff like clubs. You know, that, I don't know, these look like spools or something.
Like something landed in Japan a while back.
Crazy stuff. This is basically the one you'll be seeing today, one that looks kind of like this. So that you see how they're like little glass-like
Looks
Looks like wind
spicules.
turbine.
Yeah,
I've
yeah, it
seen
does.
Mercedes Benz.
This is what is known as a glass sponge, and they have a silica skeleton. Those little spicules will join up together and make that kind of structure. When the living portion of it, that would be the mesoglia and all that stuff that's inside of it, dies away, you actually have, you ever seen a loofah sponge? It grows from a gourd, right? It's like a zucchini type of a thing. So it's kind of like that. You can end up with this kind of glassy, it's a little bit bendable, but it will break eventually, yes.
It's also kind of like fiberglass.
Yeah.
If you touch it, it will get you full of glass.
I don't know if this will do that, but it is kind of glassy like that. Now, another protein, those spicules come from a type of protein production, but some sponges don't have any spicules. Instead, they only have a protein called spongin, which is, of course, where we get the name sponge. And spongin is a web of protein which makes them soft. It makes them like this. Okay, this is like an artist sponge. No spicules inside that one. This is what we think of commercially, right? That we would use, sponge divers would go down and get these so that we could use these in the kitchen. I'll show you a picture of that here in a few minutes. Okay, feeding strategies. This is going to go in your box saying feeding strategies, okay? They're sessile. Sessile means that they don't move. When they get to the place that they find that they like and they want to live there, they will plant down on the bottom on something hard, what we call a hard substrate. And then they don't move. Some of them, interestingly, will plant themselves on the back of a creature that does move, and they'll
Like a
move along
whale.
with him. Typically, it's something that's more benthic, something that's on the bottom. Even more like
A crab.
a crab. There you go. A crab or a
strap.
scallop. I've seen them on top of a scallop before. So they are filter feeders. That's what you're writing in feeding strategies. They are filter feeders. Using specialized cells, the sponges draw water in laterally from the sides through those pores, remember porifera. They collect food, they exchange gases, including oxygen. They get rid of CO2. They're going through cellular respiration just like we are. They're breathing, if you will, just underwater. Okay? The water is drawn in laterally, exit through the top. That's called the osculum at the top. Some sponges, it's a little more obvious than others. When I look at one like this, it's probably cut from a larger one, you know, so I'm not sure exactly where the osculum is. This isn't the whole sponge. It may have been a big one that had a big top coming out like that. I have some whole ones. There's a whole one.
Whoa.
That's you.
And
Can I hold it?
we're gonna, at lab time, I'm gonna hand all these out to you.
Can I eat it?
No, you may not eat it. So the special structures, this is kind of a cartoon version of some of the really cool cells going on in this very simple life form that turns out to be super complex. How about that? God is saying, yahoo, showing off again, right?
Clog, right?
Say again?
That's like the blood clot, right?
Oh yeah. It just is, it's incredibly complex, even though it's supposed to be simple. This first thing I'm pointing to is a little cell inside the mesoglia right here called an amoebocyte. We know about amoebas, right? We've looked at them under the microscope. They have that amoeboid movement, right? They don't have any particular shape. They actually live inside the mesoglia right here inside there. Yes?
Do you want us to write this as well inside special structures?
Yeah, put that in special structures.
Oh, well.
Amoebocytes, they are the thing that actually ingests the plankton that's being pulled in through the sides of the sponge. They do the work of eating. Some people think that they have, joined in later in an evolutionary pattern. So I think it was designed that way to have this little eating cell inside, sort of like we have a stomach, right? That's their form of a stomach. We're going to see here the amoebocytes move through the sponge using pseudopods. They can move around. Working along with, write that down, collar cells. Collar cells are amazing. They're super cool. What they do is they have a little flagellum. Remember that little tail like bacteria? And they're just spinning those around. And as they spin those flagella around, it pulls the water in to the sponge. See, they're inside here and they're pulling the water this way through those fancy collar cells. And they're not just making that motion to get the water coming through. Remember I told you about nature, really, suction's not really a thing, right? So this is just the motion of the water created by that energy being put into these flagella. So I'm going to come in closer to some of these. This is a spicule, by the way, sticking out right there. I'm not exactly sure what it looks like something from a computer, like a microchip kind of a thing, yeah. Not sure what that's supposed to represent right there. Okay, so let's take a look. Here's our collar cell. So look how the amoebocyte is right up next to it. The collar cell is whipping its flagellum around, and look, it gets in these little mucousy areas of the collar of the cell. That's where the food particles get stuck in there, and it passes through the collar cell, and the amoebocyte just hangs around the other side and says, ooh, this is a delivery window for the food into the amoebocyte. That's pretty complex. All right, they come and digest it right then. So pretty good. That's called a cholandocyte right there. We don't need to know all those words, but we understand how a flagellum works. Okay, so we can see these different shapes of him. Here's one that has a whole lot of spicule action going on. That's why it can be so tall. We can have tall sponges as long as the wave action is not too heavy. If it's too violent of waves, it'll just knock them over, right? So they have to be in a calmer area. They tend to be associated closely with coral reefs and things like that because it's calmer in the coral reef area. Furthermore, some of them have algae living with them, sort of like we talked about that with the zooxanthellae, right? And so those would live with them and perform photosynthesis to help with something like that. They need to be in the sun. So they need to be in the photic area, the sunny area, and they need to be in calmish waters, usually warmer rather than cooler. Here's a big sponge, right? A barrel sponge. When we see a fish maybe hanging out in there like a cave, but the pores are here. That's the osculum, and the pores are all on the side. Here's a pretty big one. A diver's coming close to one. Divers have gone down and injected like a very fluorescent type dye into the bottom, and you can see it being pulled through and out the top. So you can see the flow of it taking through there. Question?
Have you been scuba diving?
I have, yeah.
That's not surprising.
Here's a pectin sponge attached to a scallop. There's that scallop with his eyes. Here's the bottom side of it. But that's
They
the
have
pectin
eyes.
sponge. Yeah, they have tons of eyes along there. Oh, we haven't talked about these guys. That's right.
Creepy.
We'll talk about scallops soon. I'm going to cook some of them for my marine biology class. I'm going to eat
Yum.
it. Reproduction. Is that your next box? Reproduction. They can reproduce asexually through a process of budding. That's just like bacteria. We'll just take one cell and turn into two cells. We call that budding. They can do regeneration. You can cut a sponge into a bunch of pieces and it's called aggregation. They'll just grow new sponges. Pretty cool. Then there's something called a gemule. You're going to be drawing this today in the microscope. It's a cluster of cells encased in a hard spicule-enforced cell similar to a spore. That is one of their ways of preserving in times of, if it's tough times, if, for instance, if the tide has gone really, really low for some reason and they're out of the water for some reason. Sponges don't like to be out of the water. But if that happens and they start to dry out, they will form a gemmule, and the gemmule will be able to be planted in a new place to form a new animal.
Why did you say planted air quotes?
Well, because I don't want it to seem like I'm talking about a plant.
Mm.
When I say plant, it's going to be sown,
Yeah.
but not a seed. It's a gemmule. It's a gamete, or it's kind of like a spore. Okay. Sexually reproduction would go gametes released into the water. We call it broadcast spawning, where a sponge will just release all of the sperm or the egg, and then just hope that it meets up with a sperm or an egg somewhere and makes a new sponge. So that can happen as well. All right, so here we go. Here's a little bud forming on the side, and it'll just pop off and just form its own little sponge right there. Okay, that's asexual reproduction. Here is spawning, so it's releasing all of those gametes into the ocean to meet up with the gamete of the other type, right?
It gets smoky.
Yeah, it does. And there are certain places that scuba divers will go specifically to a place to watch the spawning of the sponges. They only do it like twice a year.
Spawning.
And when we look at corals, the corals only do it once a year in the Great Barrier Reef. So divers go down and try to catch it on camera. All right, commercial use. Look, these are all sponges, okay? In the old days, we had people called sponge divers that would dive down and go cut the sponges off. They had to go fairly deep, so these people had learned to hold their breath. You've heard of pearl divers, right? Going down to get the shellfish that has a pearl in it. Similar to that. And so back in the day, before we had like chemistry making the sponges we use now in our sinks, those are made in a chemistry lab.
Daddies and all that.
Yeah. We used to use these sponges in our kitchens, all right, because they have pores in them, they hold water, that kind of thing. The ones we fabricate in a laboratory actually do a better job, though, and they're cheaper, yeah, but they're a bunch of chemicals. All right, we're going to move on to something called phylum Cnidaria or Cnidaria, however you want to pronounce it right here. These are called the stingers. I have the underlined. Does that mean you have to fill in that blank?
Yeah.
Okay, the stingers, they have something called nematocysts. We'll cover that in quite a bit of detail here. A nematocyst is the way that they sting you. It's pretty fascinating. Has anybody ever been stung by a jellyfish? No fun, yeah?
Yeah, I get stung at Port A.
Yeah, the ones that we have here in Texas, they hurt, but they don't kill you. So,
Yeah,
but they do hurt.
We went down not realizing it was jellyfish season. We went down during it was after that big storm.
We'll wash ashore. They'll be on the
There
beach.
was a few like man of wars, whatever they're called, on the shore.
At
On the
Portuguese
shore.
man of war? Yeah.
Yeah, I know. Some of those you're like hospital or whatever. We were in the water and I was swimming with some of my friends and out of nowhere, I was like standing out of nowhere, I just felt like right across my chest. And like for 3 weeks I had a big old red, just like it was like 2 like lines.
Yikes.
It hurt so bad.
Yeah, they hurt. Yes.
Can I get a box jellyfish and keep it as a pet?
No. Don't do it.
Question,
Yes.
can I get a stingray?
Keep it in
We're back to the trying to keep a
Husky version.
saltwater tank the right salinity. It's hard to do. Go down to the Texas aquarium, and you can pet them there. So,
And get
Time
salt?
All right.
Ocean came up once.
the anatomy. Do you have boxes for anatomy for Cnidaria? Okay. The anatomy. They have two major forms: the medusa form and the polyp form.
Is there anatomy you said?
Under anatomy, correct. So the polyp is a sessile form, kind of like sessile membrane not moving. It's going to plant itself. Its tentacles are facing up. Whereas the medusa form, Medusa, you know who Medusa is in Greek mythology,
Snake
right?
rays.
She's got the snake hair. So that's the typical bell jellyfish where the bell's on top and the tentacles are hanging down below. We call that Medusa. It's free swimming. When I say free swimming, they are considered plankton. Plankton means they're floating.
Don't they
Now,
just get pushed
they
around
can,
by the water?
they can swim against, but they can't swim against the current. So when you got that jellyfish sting, he just bumped into you. It wasn't like he was
Yeah.
chasing after
No.
you. Yeah. So but if you put them in a
Because, like-
tank with no current in there,
Yeah.
they can swim beautifully.
Yeah, then after that happened, we realized when we went like into like the high current area, we would see like groups of them like going by.
And then just kind of avoid them.
Well, because how it works is the current of Port A is really strong. You know, when you go far out, you get pushed this way.
Yeah.
And like when you go out, you don't realize that the water just slowly moves you. We ended up in one of like the flaggers. It's like this is a jellyfish zone. You're not going here. We got pushed into it, not realizing, and so that's why we got stung. I got stung. Malachi Gonzalez got stung, and like I think Hannah got stung too.
Okay. Examples would be jellyfish, sea anemone, hydra. We will be drawing a hydra today in the microscope, and we'll be looking at corals today as well, just not in the microscope.
Oh, hey.
Anatomy continued. All members of Cnidaria have outer cells called epithelium, mesoglia, jelly-like substance. That sounds a whole lot like the sponge. They have radial symmetry and tentacles. Remember we said radial symmetry, a good example is sea anemone, and that's in this group right here. The tentacles have something called nematocyst. That's a special structure. So go ahead and write for special structure nematocyst. That's the thing that stings you. They release toxins to sting their prey or predators if they're trying to get away. Some of the toxins can be really, really bad, like kill you bad. That's the box.
I like the men of war. Don't those kill you?
The Portuguese man of war, you know, those are like hundreds of jellyfish all together in a colony. It's a colony of jellyfish.
Then what are those little?
Each tentacle is an individual.
Then what are those little blue ones that wash on the
With
beach?
yeah, those are them. That's it. Yeah,
those-
yeah. If enough of them sting you, you could, especially if you have a sensitivity to them.
We
Go
found
into
one. Me and my
anaphylaxis.
uncle found one on the beach.
Yeah,
It
they're
was
pretty.
crazy. Yeah, we went and like buried it in the sand
Okay,
and stuff.
there you go. Yeah, I wouldn't even try to touch it, even if it's
We
dead.
had like a little show.
They can sometimes still release their nematocysts even if they're dead. Yeah.
I don't like how snakes, if they're dead, they can still bite you. If they're like
Well, the
recently
venom
dead.
is still there. Right.
Yeah.
So if it can get to you some way, if they're that really just dead, then they can actually bite still. Okay, we're gonna watch a video.
Some are longer than a blue whale. Others are barely larger than a grain of sand. One species unleashes one of the most deadly venoms on Earth. Another holds a secret that's behind some of the greatest breakthroughs in biology. They've inhabited the ocean for at least 500 million years, and they're still flourishing as the sea changes around them. Jellyfish are soft-bodied sea creatures that aren't really fish. They're part of a diverse team of gelatinous zooplankton. Zooplankton being animals that drift in the ocean. There are more than 1,000 species of jellyfish, and many others that are often mistaken for them. A noted feature of jellyfish is a translucent bell made of a soft, delicate material called mesoglia. Sandwiched between two layers of skin, the mesoglia is more than 95% water, held together by protein fibers. The jellyfish can contract and relax their bells to propel themselves. They don't have a brain or a spinal cord, but a neural net around the bell's inner margin forms a rudimentary nervous system that can sense the ocean's currents and the touch of other animals. Jellyfish don't have typical digestive systems either. These gelatinous carnivores consume plankton and other small sea creatures through a hole in the underside of their bells. The nutrients are absorbed by an inner layer of cells, with waste excreted back through their mouths. But the jellyfish's relatively simple anatomy doesn't prevent it from having some remarkable abilities. One kind of box jellyfish has 24 eyes. Scientists think it can see color and form images within its simple nervous system. 4 of its eyes are curved upward on stalks. This allows the jellyfish to peer through the surface of the water, looking for the canopy of the mangrove trees where it feeds. In fact, this may be one of the only creatures with a 360-degree view of its environment. The jellyfish's sting, which helps it capture prey and defend itself, is its most infamous calling card. In the jelly's epidermis, cells called nematocysts lie coiled like poisonous harpoons. When they're triggered by contact, they shoot with an explosive force. It exerts over 550 times the pressure of Mike Tyson's strongest punch to inject venom into the victim. Some jellyfish stings barely tingle, but others cause severe skin damage. The venom of one box jellyfish can kill a human in under 5 minutes, making it one of the most potent poisons of any animal in the world. Other jellyfish superpowers are less lethal. One species of jellyfish glows green when it's agitated, mostly thanks to a biofluorescent compound called green fluorescent protein, or GFP. Scientists isolated the gene for GFP and figured out how to insert it into the DNA of other cells. There, it acts like a biochemical beacon, marking genetic modifications or revealing the path of critical molecules. Scientists have used the glow of GFP to watch cancer cells proliferate, track the development of Alzheimer's, and illuminate countless other biological processes. Developing the tools and techniques from GFP has netted 3 scientists a Nobel Prize in 2008 and another 3 in 2014. But it's jellyfish who may be the most successful organisms on Earth. Ancient fossils prove that jellyfish have inhabited the seas for at least 500 million years, and maybe go back over 700 million. That's longer than any other multi-organ animal. And as other marine animals are struggling to survive in warmer and more acidic oceans, the jellyfish are thriving, and perhaps getting even more numerous. It doesn't hurt that some can lay as many as 45,000 eggs in a single night. And there's some jellyfish whose survival strategy almost sounds like science fiction. When the immortal jellyfish is sick, aging, or under stress, its struggling cells can change their identity. The tiny bell and tentacles deteriorate and turn into an immature polyp that spawns brand new clones of the parent. As far as we know, these are the only animals who've found a loophole when facing mortality. That's pretty sophisticated for species that are 95% water and predate the dinosaurs.
Pretty cool stuff. Okay, so here's some that are glowing. They are probably have, they do perform luminescence, but sometimes it's just we're shining a black light on them and they glow. Here's the box jelly right there. I don't know if we can see those, whatever number of eyes they said about it. These, of course, are very common in Australia, naturally, where all the stinging things live. Here is the long one, the big one, the lion mane one. Oh, and this, I just want you to see, these are ones that are swimming in a tank with no current, and you can see how graceful looking they are as they just move along. Yeah, there's no sound here. They're just real pretty with their tentacles hanging. It'd be fun to have them as a pet. Very cool. Yeah.
One of my aunts has a saltwater tank with a jellyfish in it.
Wow, super
It
cool.
just does circles around the
Would
tank
you-
for hours.
Rather be a fish
Poor thing.
or a jellyfish?
A human.
I want to be the
Gotta be a
immortal
human,
jellyfish.
yeah. Reproduction in Phylum Nadaria, they have budding and regeneration. We're going to see, this is a critter we're going to look at under the microscope called a hydra. That budding is famous for budding, and the one you'll be looking at, in fact, has a bud on it. Sexually, they produce gametes expelled in the water through broadcast spawning. The jellyfish cycle can be very interesting when they have both the polyp and medusa stages.
So what do they do for reproduction?
Budding, regeneration,
Yeah.
or gamete production, of what we call broadcast spawning. But when he mentioned in that video about the fact that when they grow old, they can kind of spawn off a new critter there, we do see that in their life cycle. Which I'll
So
show
are you telling me there could be like a jellyfish 7,000 years old?
No, because it's a clone. It's not the same jellyfish.
It's a clone of the same one.
A
No,
lot
but
of them
it's a
don't,
clone.
make it to adulthood because all the babies get eaten by something. So
Nice.
not necessarily do that. All right, so here we'll see that they would be a small creature like this in its polyp stage, but as it gets bigger and bigger, look at the polyp starts budding, and look, this top part peels off, turns upside down, becomes the bell, and here we have the medusa stage right here. Okay, we have gametes coming for sexual reproduction, forming an egg and something called a planula. And that planula will implant itself and it'll start over. So in other words, it'll go through both the medusa and polyp stage.
That's pretty cool.
So they're really neat creatures. The nematocyst is crazy. Here's some actual photographs of a nematocyst that is discharging. They're wrapped up in a little capsule with a little trigger hair right there. When the trigger gets knocked, what happens is water fills this, and it sends that out, and that thing will stick into you. So I think this is showing a picture of one coming out. Here you can see here's a good picture of it right here. The tentacle sending that out, it's kind of coiled up in here and it shoots out. Boom. And a lot of times they will stay in your body. That's why they're hurting you. Here we can see it's going to open up the little door. The water's going to rush in, cool it down, and cause that to swell up and boink, just like that. Okay, we're going to watch another video, just because it's too fun.
Do they just replace themselves after they're gone?
Good question.
You're swimming in the ocean when something brushes your leg. When the tingling sets in, you realize you've been stung by a jellyfish. How do these beautiful gelatinous creatures pack such a painful punch? Jellyfish are soft because they are 95% water and are mostly made of a translucent gel-like substance called mesoglea. With such delicate bodies, they rely on thousands of venom-containing stinging cells called cnidocytes for protection and prey capture. Even baby jellyfish, the size of a pencil eraser, have the ability to sting. Larval jellyfish, Ephyrae, look like tiny flowers pulsating in the sea. As they grow, they become umbrella-shaped with a bell at the top and descending tentacles around the margin. The largest species of jellyfish, the lion's mane, has tentacles that can extend more than 100 feet, longer than a blue whale.
That is crazy.
These tentacles contain most of the stinging cells, although some species have them on their bells too. Venom is ejected via a nematocyst, a whip-like hollow tubule, which lies coiled under high osmotic pressure. When mechanical or chemical stimuli activate an external trigger, the lid of the cell pops open and seawater rushes in. This forces a microscopic barbed harpoon to shoot out, penetrate and inject venom into its victim. Nematocyst discharge can occur in less than a millionth of a second, making it one of nature's fastest biomechanical processes. Nematocysts can continue to fire even after a jellyfish has died, so it's important to remove lingering tentacles stuck to the skin. Rinsing with vinegar will usually render undischarged nematocysts inactive. Seawater can also help remove residual nematocysts, but don't use fresh water, because any change in salt balance alters the osmotic pressure outside of the cnidocyte and will trigger the nematocyst to fire. That's why urinating on the affected area, a common folk remedy, may do more harm than good depending on the composition of the urine.
That would be funny.
Most jellyfish stings are a painful nuisance, but some can be deadly. An Indo-Pacific box jelly, also called a sea wasp, releases venom which can cause contraction of the heart muscles and rapid death in large doses. There's an anti-venom, but the venom is fast-acting, so you need immediate medical intervention. Despite the impressive power in their tentacles, jellies aren't invincible. Their stinging cells are no match for the armor of thick-skinned predators like the leatherback turtle and ocean sunfish. These predators both have adaptations that prevent slippery jellyfish from escaping after they are engulfed. Backwards pointing spines in the turtle's mouth and esophagus, and recurved teeth behind the sunfish's cheeks. Even tiny slipper lobster larvae can cling to the bell of a jellyfish and hitch a ride, snacking on the jelly while they preserve their own energy for growth. Small agile fish use the jellies as moving reefs for protection, darting between tentacles without ever touching them. Nudibranchs, which are sea slugs covered in protective slime, can actually steal the jelly's defenses by eating the cnidocytes and transferring them to specialized sacs for later use, as weapons against their own predators. Even humans might benefit from the sting of a jellyfish one day. Scientists are working on manipulating cnidocytes to deliver medicine, with nematocysts rarely 3% of the size of a typical syringe needle. So, the next time you're out in the ocean, be careful, but also take a second to marvel at its wonders.
All right.
So wait, so it can take them and then use it for
The
itself?
nudibranch can, yeah.
That is
Yeah.
cool.
So this is another kind of a cnidaria right here called the hydra. We'll be looking at that, and you can see its tentacles are on the top, so it's sort of in the polyp form. And they can bud off at the bottom right here. Gastric region would be where their stomach is. And if we look at the cells inside, you can see once again, look at these, look how similar that is to the sponge with the little interesting flagellum right there, which does pull water through. Okay, and then we have our mesoglea inside, and then our ectoderm, the inside of the cells right there.
You know, there is a myth about the hydra as well.
Oh yeah, I'm gonna tell you about it right now. So here's the deal about it, since it buds like that, the myth, what's the myth of the hydra?
3 more come back, right?
Yeah, you cut off a head,
It's a really scary snake dragon.
Dragon thing.
And it was
There it is.
spit, I think it was acid.
okay, and you cut the head and 2 grow back, or 10?
3.
3? Okay. Yeah, and so that's the same here with our hydra right here. You cut them, they're gonna make more. So that's part of their reproduction, right? That's the budding, and that's the just aggregate. They can aggregate and make new ones. All right, the sea anemone is in this group too. These are the tentacles right here. Here we know the story of the clownfish and the sea anemone, right? The clownfish is lacking a certain enzyme that allows it to get stung. That brings us to coral. Marine animals that live in large colonies. This is another example of Cnidaria, would be coral. The tentacles surround the mouth, so you may not know this because they're so small. They're like little teeny tiny sea anemone, is what coral look like. The creature makes an exoskeleton of calcium-rich material that they form like a hard substrate. They make the reef. An incredible amount of beautiful diversity in this group. See, it looks like a little tiny sea anemone. Here, when they're young, they look like this, and then they build up this little cup around them, and that cup is hard, and this part down here is hard. Old, dead corals, they build on top of them. There are obviously different kinds of coral. This is considered what we call a soft coral. These are the little tentacles. If you come up close to this one is in a feeding stage right now. If you touched it, all those little tentacles would be pulled back
Would
in
they sting
to
you?
these little sparks. Sure, but they're so small we can't feel it.
Oh,
Yeah, because they're really tiny.
What if it's super bad and it's
I can't think of a coral that is.
That
They're
looks like
just
a
all so
brain.
small. Right, this I believe, there's one called brain coral, but I don't think it's this one. Here, the next one's brain coral. But these have, there's a little holes in there, and every little hole has a little creature living in it. Here's a brain coral. Look at that one.
kinda
Yeah.
looks like
And
A
each
brain.
one of those little dots right there is an
Man, I always have
individual.
kids.
So they live in a giant colony. Here's some, these guys have the little tentacles pulled into those little tiny holes, those little tiny pores right there. They're not feeding. This is non-feeding time. This one's called the sea fan. It is an animal. It seems weird to think of that as an animal. Yeah,
It kind of looks like a fan.
it does. That's why they call it that. Look at all the color there. So the corals have all the different photosynthetic algae, zooxanthellae living with them, giving them color like that. Here we have a swimmer near a coral reef. Some coral reefs are more colorful than others. This is called a mushroom coral. I actually have a sample of this to show you. A student bought this for me at a store, and we had to figure out what it was. But this is not a colony like the other corals are. This is an individual. This is one creature, and that is its mouth. So it pulls water through these grooves into that mouth, and it looks like that. One of them's called the sea flower or something like that, because it
Are we
looks
gonna
kind
be
of like a
able to pass
flower.
it around?
Yeah, we'll look at it during lab time. So we see here like that, there's that mouth right there. Okay, this is your matching or whatever at the bottom of your page.
Yeah.
Ready? Number 1, mesenchyme. It's a jelly-like substance that separates the epidermis from the inner cells of a sponge. We also have something very similar in the jellyfish called mesoglea that we'll get to in just a minute. Remember those collar cells and those spicules and those amoebocytes. So you're matching number 1. You got it?
Yeah, bottom.
Great. Oh, right, I circled
It's the bottom
mesenchyme,
one, by the way.
mesenchyme right there. Okay, number 2, the gemule or gemule, however you want to pronounce it. Cluster of cells encased in a hard spicule reinforced cell. Remember, it's sort of like a spore, so if times are rough, then it can survive by having this little life raft, if you will. And
second
the
bottom one.
cells are on the inside. There's a question? Got
Second
number 2?
bottom one.
A second at the bottom, okay. Number 3, amoebocytes, the cells that move using pseudopods and perform different functions. We know that they have them in the sponges for sure. It's this red one right here, the amoebocyte. Number 4, collar cells. There's that beautiful collar cell, the flagellated cells that push water through the sponge or pull water through the sponge. They also capture food in that little mucus collar right there. Number 5, the polyp is the tubular form of a cnidarian with a mouth and tentacles on one side and a basal disc to stick to the other side down. So this is the polyp, is the one that's got its tentacles upward. Medusa is a free swimming cnidarian with a bell-shaped body, and typically its tentacles are downward. That's a cool looking jellyfish right there. Mesoglea, the jelly-like substance separating epithelial cells of a cnidarian. This is somebody holding up a jellyfish that's tentacles are gone or whatever. Maybe they dissected it. Number 8, nematocys, small capsules that contain a toxin that's injected into the prey. We saw how that works. And now it's time for lab.
Get out of here. I had it right.
So, here's what we're going to do, y'all. You're going to look at sponge spicules. They're in this scope right here. I only have one slide. And you're going to look at it in 2 different magnifications. Whatever the 2 look like. You have 2 spots to draw that. Do you understand what I just said?
Yep.
Yes, ma'am.
Spicules, you're drawing 2 pictures of one slide in different magnification. Hydra, I've got a ton of those. I'll be bringing those to the rest of the scopes. Spongilla, gemule, I only have 1 of those. And then we're going to be drawing planaria. You're going to turn over to the next week's lab to draw the planaria because I don't want to bring the microscopes with me next week because we're going to dissect a earthworm. Okay? So go ahead and start on that one. And I'm going to bring you the, Hayden, get up and bring these.
Wait, how
So
do
that
you
on
use
page
the
99.
earthworm?
Take these over
I don't
to
know how
the
use
slides.
earthworms for fishing. Is it like this one?
Same one.
All the rest of the scopes. 1, 2, 3 scopes.
Oh my.
So we're drawing 2 of the sponge spicules, 1 of the hydra, 1 of the spongilla gemule, and 1
So-
planaria.
1 hydra, then 1.
Are those hydra, Hayden, those are the hydra, right?
Yes. This
All right, so I'm going to bring to these
no.
the gemule on this way too. So y'all have the only slide for gemule and spicule. And then this hydra looks a little lame. It might be all right.
Don't talk to it like that. I don't
It's
feel
Sponge
it.
pickle on this page. Sponge pickle on this page.
The other ones I can actually see.
One of them first. The 2 of them on this page.
Okay, so you want us to do the hydra?
Yeah, in your book it says on page 97 in my book. I don't know what about your book. It says spicules. It has 2 spots for
I
it.
have a hydra.
Then who's this?
And then the next page says hydrate. It says jellyfish, but you're going to use a spongilla gemule instead there. We're not drawing a jellyfish.
That is cool.
So weird.
Is that the hydra? Yeah, you'll be
Yeah.
drawing that on page 98. Okay, that's my
That
page.
is cool.
Yep, I'm gonna use these.
Okay, while you're waiting, which one?
I can't find any.
Which one are you looking at?
Hey, you zoomed out.
But I already had them in focus.
Oh,
What are you doing?
Put them in there earlier.
Why was it so cool if you had it?
That one's cool looking.
Is that the hydra?
Yeah.
Hydras are awesome. So, I'm going to bring some stuff around for you since you're not looking at something. Because that is that mushroom coral thing. Here's some other corals. Notice they're what we call bleached because the little corals have their color because they
Oh my
have
gosh, I lost it.
algae living in them. So when they die, they turn white. This one though is built itself out of a different
He's a
color,
better
so
friend
that's
than
why
me.
it's dark. Okay, and remember you're gonna look at it in 2
Oh, you
Different.
betrayed my friendship.
magnifications.
Yeah, but
We'll let it go.
What?
Go ahead.
You're
Look at it.
look at his, bro. He's
10.
on the farthest out, so
Whatever,
he
100.
is.
40, that's probably 40 actually.
Oh my gosh.
I had to go ahead and write down that was 40x.
40x?
The lowest thing is 40x. Oh, for the hydrate, then whichever one you like the best.
This one.
Look at this, look at this. Look at
Hydras are very cool. Oh, it's super cool. Oh, look at his tentacles. Awesome. Oh, he's so cute. I like it. He fits all the way in there. Okay, which one did you look at? That one?
That one. The one that Judah has.
Okay, while you're waiting for another slide, here's some different corals. Notice those little holes?
Yeah.
A little creature will live down in those holes. And they don't have color
Do
because
you
they
have a
don't have any
flat,
algae.
clear one that we could look at under a microscope?
No. That's why we're just looking at sponges and stuff. And then I also have these different sponges. These are all different. I just want you to touch these and feel how some of these are really hard.
Can we hold the sponges down here? Some of the big ones?
Yeah.
oh, I need to look at
Terrible drill.
presentation.
Some of them are cut.
I mean, two or
for you to take a look at.
Dude, you actually got a photo of it. That's dope.
These are all sponges.
I feel like that would never break if I touch it.
Maybe.
I don't know.
This one is the only one that's
It's
mostly
like
sponge, and the
12
rest of them have
tries to
spicules
look
in
at
them.
it.
Really cool spicules.
I'll take a picture of this one too. This one's like a full one.
Okay, you have two of this? Two different views of this one?
Of the sponge she has it.
Okay, did you look at both of these?
I looked at that.
Which one's this?
The Hydra.
Oh, is this a good Hydra?
I haven't looked at it.
Alright, where are
Okay,
the
I didn't kill-
sponges? You want us to look at
Then
this
we're gonna
under a
put that one
microscope?
here.
No,
I
you
gotta
just bring a
send
look at
that
it. There's
on.
also these.
Whoa.
You want us to look at these under the microscope?
No, no, you just look at it with your eyes. These are some other sponges right here,
Look at that one.
and of course we have these sponges.
I think this one's
These
super
are
cool.
corals. Those are the spicules, those are coming. You're gonna draw that there, and here you're gonna draw the gemule, but you only have one slide, those are over
Okay,
there.
okay.
Right now, you can, I just want you to use your observation skills to look at some corals, some sponges, these are also
is
sponges,
cool.
all we need to see is
Dude,
some
that's
sponges are
editing.
hard,
That's impressive.
they're not squishy.
Like these will activate with water, right?
Well, no, they're dead,
Oh.
but when they were alive, they would.
Look at that.
Okay.
Yeah, there's nothing left.
So are there
Coral?
certain
Yeah.
sponges that would?
There's also
If they have
that
a
blue
gemule,
one.
one of
is
those
pretty cool.
gemules,
Yeah,
yeah.
just
Oh,
push
that's
that
cool.
microscope
That
down.
looks like a
Okay.
boxing glove.
Right, yeah, when they pull them up, they
Alright,
would.
good. That's dope.
Alright,
So
and
wait,
also
so
you're
what are we looking
waiting.
now? What are we looking now?
You're gonna just observe these, but I'm gonna bring you a planarian to look under there at.
Planarian.
But it's for next week, so you'll just have to draw it on the back of the page.
No,
Those
there's
are
a
coming
section
from
for
over
it
here.
over here, planarian and tapeworm.
On the next lab, right?
Yeah.
Yeah, I want you to draw a planarian there. And I've got several, and they're huge, look, you
So
can
what,
see them.
what if I were to lick this?
Well,
I don't know. My brain's
I
not-
need-
there's germs on
I
there.
need a microscope one.
Dude, don't lick it.
I'm going to.
Why does it look like that?
Well, for some reason, they all
I- I'd
congregate
say
into the
too.
bottom, I'm not sure.
No,
Doesn't
like,
taste
where
like
did
They
it
glue
go?
it on.
Nothing.
That's one of the professional techniques. Have you looked at that one yet? No.
It smells.
Okay. This is
Smells
the
like- it
spicules.
smells like a beach
Y'all
house.
all three looked at the spicules. Tori, did you look at the
Surprise,
spicules?
surprise.
Yes. You did.
No, it smells like a beach house.
All right, y'all, these are the spicules. I only have one of these slides, okay, and you're gonna draw it under two different magnifications,
This is a
okay?
Oh.
That's a coral.
Oh,
These are sponges, these are corals, the hard things are corals. Look at the
okay.
coral
I
here.
thought I've been
So
holding
dumb.
sponges this whole
These
time.
are sponges, these are all sponges. You're gonna look at a planaria in a minute. Yeah, these are, look how hard this sponge is, very spicular.
Yeah. Is this cool?
That's cool.
Blue
Not
though.
the microscope.
Yeah, it's made
Here's-
it's
you can
substrate
look
out
at
of something hard.
the planarian over here,
Look
Eddie,
at this sponge.
for the next page.
This is suspicious.
That's for the next lab.
Yeah.
Squeeze that one, that's got a bunch of spicules in it.
This is
See, really
squeeze.
hard.
It looks like a piece of cactus.
Yep, and here's another one, also very hard.
It looks like a dice.
This one has a little bit of spicule and spongin in it.
So what is it I'm looking for here?
Okay,
Anything.
you may need some help with that one. They're not, there's no stain on these guys, cause they're kinda like glass.
Do we have a
It's
jellyfish
stain.
thing to look at?
No, we're changing it to gemule.
Oh, okay.
Okay, so you're gonna look right here, you're gonna see this thing, a bunch of little needles in there. And they don't taste.
We're all
so
adults
they're
here.
not. And then you're gonna go up one more magnification.
And that's
They look like little hairs.
Yeah,
What is this?
that's
what?
40x, and then go up to,
you can even see all the hairs and stuff on this one.
Hydra?
that's the budding hydra. Hydras
Yeah.
are super cool.
Mm-hmm.
Are
Have
these
y'all looked at
jellyfish?
hydra yet?
Yes, but which one's the jellyfish?
We're using the gemule. We're replacing the jellyfish with
Oh,
the
the
gemule.
gemule.
Yeah.
You can do it first.
This,
Or the-
you looked at the,
You know there's a sponge on the other
that's the spicules, right? You did
Yeah.
it under two magnifications, right?
and then
That's jellyfish, we're changing it to gemule.
Mrs.
A
Casper.
Sponge gemule. I wrote it up there.
Just
Spongilla
literally what it looks like,
gemule.
right?
Seriously?
Yeah.
Did y'all already look at
That's literally what it looks like.
budding
Look
hydras?
at it.
Okay, look at that. Let me see.
It's.
One to one replica.
What am I looking at here?
Look at that one.
One to one
That's the,
replica.
Oh, planaria for next time. Isn't that crazy? I'm gonna put under here, hold on. I want you to put.
Literally.
They're so big.
Oh my goodness, this is so
I wanna
short.
put.
I literally sat here just doing
Look at
this.
his face.
Just like,
I want you to look at his face. He's looking back at you.
what is it we're gonna do
See
next?
his little eye spots up there?
One-to-one replica.
Oh, I do.
Of the
Oh, that's
spicules?
crazy.
Yeah, at
And they go up
40X.
one more.
They go to
You're gonna
100.
draw, the other way.
Oh.
That's gonna be too close. There you go. Try that. It's just gonna be the same thing.
How
Just a
are
little
you
closer.
mascots if I'm correct?
Oh, that's cool.
Where'd it go? You took it out.
Here's this mushroom coral.
Mushroom coral.
Oh, they're like hollow.
Whoa. That's some weird feeling stuff.
So they're like hollow. They're like.
Yeah, they're like
Big.
little glass structures. Yep, that's them. Like little needles, and you're just gonna draw that at 100x, 40x, and 100x. I don't know why they want us to draw that twice. It's not as
Eddie,
exciting
Eddie,
as the
you go
budding,
buddy.
the budding.
Hold the
plant.
mushroom coral.
Did you find a really cute little purple budding
What was
coral?
this one?
I'm looking for it.
Is this the
How's the
yellow
Daniel
one?
look?
What the
Are
heck?
they too crowded? They're
Dude,
really
It's
crowded.
looking
What is
back
it?
at you. You see the eyes?
That
I
is so
found
cool.
it. Isn't it cute?
He's so yellow.
So I would be kissing this.
Let me see.
I would be kissing
Alright,
a
so the
mushroom.
brownish
Whoa.
things
Yeah, I know, right?
are the gambier
Yeah, I
stuff
saw
all
that
around
one, but I
us,
didn't get
or
like
spicules.
really.
So the brownish colored thing is the gambule.
Whoa.
And all
what's
those
that
little
green?
needles are
That's
spicules
crazy.
all
Really
around it.
pissed
We're gonna say
off.
come from the sponge.
You see the eyes?
That's a pretty
It
cool
looks really
sponge.
pissed off.
This is where this.
So what is this one?
That looks like a.
That
These
is
are these
a
different types of sponges.
planarian.
Okay.
That's a planarian.
You saw these, Eddie?
I did.
You saw
No,
how
that's where this.
feel how
Which ones?
spiculated that one is instead of spongy.
Oh,
feels
that's-
very
oh, it's
hard.
on the other page.
That's next
So
week's.
this
yeah.
helps to
Yeah.
get an idea. It's very pokey, yeah, like a
So, do we have the SpongeBob ones yet? These ones, or do they still have them?
the gambule, that's
Yeah,
fine.
that's right. Well, I have my book, so I can get the Panera.
Just draw it on the back or something. I'm not bringing the scopes back next
Yes,
week.
ma'am.
Miss Ashley.
Sponge.
Oh, thank you. Did y'all see these different kinds of sponges?
Not the little tiny pores.
Is this our today's quiz?
Huh?
No?
Like for instance, take a look at this one and see how hard it is. And that's a sponge.
Like it looks like a
Stick.
like a stem, yeah.
Yeah.
It's fluffy.
That's the one with a lot of spongin and no spicules. The rest of them have some sort of spicule.
Eddie, can I see that?
And like this one
That
has
microscope?
spicule and sponge.
Or you're
So
still
notice
using
it's
it?
squishy,
Yeah.
but
What's
also a little
this
hard.
one called?
That is
This one, it
On the
feels like
page.
red.
yeah.
Alright, these are the, has everybody looked at the
Planarian.
gambule or
I
not?
haven't.
They're super cool looking.
PL
The golden kind of thing there is the gambule. All those little things coming in are the spicules.
Thank
Did
you.
you look at a planarian for next week?
I'll take a look at that.
Okay, so turn the page. Put a planarian in.
Dude, that's so cool.
This way I will not need
Dude, look at it. I mean, just
scopes
look at it.
next week. Find
Dude,
that guy.
look
It's
at it.
huge.
That's cool though.
There's
So
nothing
I don't even know if the light
because
can.
Look
Is it
at
on?
it.
Okay, so just move it around. Yes.
Oh my gosh, it's kind of
Where'd this
orange.
go?
Look at it.
Alright, everybody's seen my different sponges, yeah?
I haven't looked
Okay.
at the big one over there.
Okay, we'll do that. Hey, you're gonna put all these back and then we're
Is
gonna
that
go back
one?
and use A, B, C, D,
That's
E,
the
F.
spicules that are
Oh, yeah.
Wow, that was pretty impressive.
That was.
Hey,
Far.
hey.
It flipped.
Well, how dare you throw it back?
It flipped pretty far.
That one went pretty high.
So these are corals.
Eddie,
Right,
flip
every
on.
one of those little holes, a little critter lived in, a little tiny skins in. And in here,
Dude,
are
how'd
all
you
those
mess?
Epic fail.
little pores.
Hey, that's blinking. That's not the
There
eye.
you go.
Have y'all all saw this?
Dude, that was impressive.
Hey,
Okay, y'all are just
contrast.
waiting for the gambule over here?
Yeah.
It's super cool looking.
Where is it?
It's coming.
Oh, okay.
Yeah,
Judah picked a field
I'm
goal.
just gonna, just leave it,
We'll
that was
just take the
impressive.
Yeah,
Sophie, just leave it right there and let's
Yeah.
take the whole scope over there. Okay, when you're ready. Here, and then you're all gonna draw one planaria that goes on next week's lab. Could you find his little face with his little eye spots?
Yes.
Kinda looking
This is
back
my
at you.
We
Planaria.
don't need a tapeworm, right?
Well,
That's
I
next
don't
week.
have a tapeworm. And I'm happy to say I do not have.
Eddie, it's one replica, right? It's exactly the same. Right? Do you think it looks the same?
Ooh, awesome. Okay, guys, here, this
Yeah, I
is
do
this
that.
is the gambule from a sponge, and if you kind of see a golden cell in there, and all the stuff around it are spicules.
Oh my
It's
goodness.
super cool. We're done with all this.
What is
yeah?
this
Yes,
called?
ma'am.
What is this called?
This is-
That's
this is-
the gambule
sorry.
Yes, where the jellyfish thing says.
Oh, that looks like.
That's cool,
Did you get
right?
that planaria?
Yeah, that's cool.
All right, Hayden, I mixed up the scopes a little bit,
Ooh.
but they have it written on the side.
Remember, hey, this is what you're getting paid for.
I wish. I mean, I didn't get any different way.
Yes, the experience is worth it, right? Goes on your resume.
Is it the 40X?
I don't know. But it's cool looking.
Yeah, and those all the things coming in around it are spicules.
Of this picture too.
Dude.
Oh my gosh. It's cool because like it almost looks like gunk at the bottom of a tank, but then
Yeah.
I realize that's super small.
Yeah, that's all in the ocean when you're swimming. All right,
And
once
drinking.
y'all all have that stuff drawn, that's it. You're free to go.
Oh, okay. Thank you, Mrs. Casper.
You are welcome. I will see you next week.
Will we get to eat the crawfish?
No.
What about the worm?
The what?
What about the worm?
No, they're all preserved, hun. They're gross. Gross. Thank you.
Yeah, a chair. I think it was a chair.
See you next week, Miss Casper.
All right, very good. Have a good week.
What are these water bottles and octopuses for?
If you squeeze it really hard, you'll see the creature sink, which doesn't seem like it ought to. You have to squeeze it really hard, really hard. 2 hands.
Thank you. Miss Casper.
Maybe get a guy to do it, not
I
to
was
be
good.
rude.
I'll try.
If you squeeze that big bottle, Judah, with both hands, you'll see the little creature sink to the bottom.
Oh, that's cool.
Yeah, it's a pressure differentiation.
Check it.
Honestly,
With
well, you're strong. Yeah, I can't do it with one hand. Sometimes I can't even do it with 2 hands.
With one hand.
I like that one because he's got a little mask and snorkel.
Oh, 2 fingers? 3 fingers?
Oh wow, strong.
It's sucking. It's sucking.
Thank you, Miss Casper.
You betcha. See you tomorrow.
Bye, Mrs. Casper.
Bye. See you next week. Have a good week.
Bye, Miss Casper.
So long. See you next week.